Hello, I'm Dr. Mark Darcy. Welcome to another episode of Science on the Edge. Today, we're going to talk about how nerves fire. So, our neurons actually fire and transmit signals around the body and the brain. So, let's get started. Okay, so neurons and synapses. By the way, if any of you want to follow me, my Twitter handle is at Mark of the D. That's at Mark of the D. Quite a few controversial tweets there, but if you're not too sensitive, I think you'll enjoy following me on Twitter. There's quite a lot there about science and technology, but a little bit of philosophy and politics as well. That's the controversial side of things. Okay, so today we're going to talk about different types of neurons, how nerve impulses are conducted around the body, and how nerve impulses pass from one neuron, that is nerve, to another at the synapses. So neurons are cells within the nervous system that transmit information to other nerve cells, or to muscle, or to glands. So nerves, yes, they connect around the brain and around the spinal cord, but also nerves connect to and stimulate muscles and glands. What we have here is a generalized diagram of a nerve. So at one end, we have the dendrite. At the other, we have the axon terminal. So the signal originates, is received at the dendrite. Then the signal will then transmit through the axon. Axons can be quite long, by the way. A nerve, for example, could begin at the tip of your finger and could go all the way up to your spine. So the axons can be very long. And then when the signal gets to the end of the axon, it reaches the axon terminal, where some sort of neurotransmitter may be released. So acetylcholine, if this is a motor neuron stimulating a muscle to contract, for example, or if this is in the brain, this could be a neurotransmitter such as serotonin or dopamine, which are involved in various sensations and emotions. Now, as you can see along the axon, we have these special cells called Schwann cells, which sort of encircle parts of the axon. They secrete a compound called myelin, which covers the axon, provides a kind of insulating layer like the plastic around a wire. Let's go into more detail about that, actually. So as we can see, there's a Schwann cell around an axon. Now, myelin is an insulating layer or sheath that forms around nerves, including those in the brain and spinal cord. It's made up of protein and fatty substances. This myelin sheath allows electrical impulses to transmit quickly and efficiently along the nerve cells. So we have bundles of axons sort of joining together to form nerves. So an individual neuron will quite often bundle together with other neurons protected by connective tissue. Connective tissue, by the way, is what also protects your muscles and other tissues in the body. And these bundles will be known as nerves. Now, the actual nervous impulse sort of jumps along the nerve at what we call the nodes of Ranvier, these little spots between the myelinated parts of the axon. Now, there are different categories of nerves. You can categorize them based on their structure, what they look like, or their function. So we can structurally classify them depending on whether they are multipolar, bipolar, or unipolar. A multipolar neuron is a neuron that basically has got all the dendrites and the cell body at one end and the axon terminal at the other. So the cell body is at one end, the axon terminals are at the other. This is the vast majority of neurons. Then we have bipolar, which basically have the cell body in the middle. Still dendrites at one end, axon terminals at the other, but the cell body is in the middle. These type of cells tend to be specialized. For example, the neurons in your eye that detect light and those in your ears that detect sound. Then we have unipolar neurons. Unipolar neurons have the cell body out to the side. Tends to be sensory neurons, such as the sensory neurons in various parts of your body that detect things like heat and pressure and various chemicals in your bloodstream. They tend to be unipolar. Functional classifications, though. Neurons are divided into sensory neurons, intermediate neurons, known as interneurons, and motor neurons. Sensory neurons transmit nerve impulses from the receptors, so wherever the receptors are, for example, heat receptors in your skin, or pressure receptors, or taste or smell, or whatever. Sensory neurons transmit their impulses to the central nervous system. The central nervous system is basically the nerves of the brain and the spinal cord. Interneurons transmit nerve impulses within the central nervous system, so from one part of the central nervous system to another. For example, the various neurons in your brain, which are communicating over time with other neurons in the brain. This is how you think and how you operate your body. Those are interneurons. Then we have motor neurons. Motor neurons transmit nerve impulses from the central nervous system to various effectors. So, for example, to the muscles, to the glands, to the heart, that sort of thing. Okay, so let's just jump through that slide. It's not really displaying very well, but it doesn't really matter. I'm going to describe neurons now in a little bit more detail. Now, the cell body contains a nucleus and other organelles, e.g. the mitochondria. Mitochondria are the powerhouses of the cell. Some neurons contain dendrons, only sensory neurons, as it happens, that conduct nerve impulses towards the cell body. The axon conducts nerve impulses away from the cell body. Axon branches, and each terminal branch ends in a synaptic knob where it connects with the next neuron. In the peripheral nervous system, that's all the nerves that are outside the brain and the spinal cord. The axon and the dendron are myelinated to increase the speed of transmission of a nerve impulse. So our sensory and motor neurons, similar in structure, both contain an axon, dendrites, and a cell body containing a nucleus. That's where the DNA is. Both are myelinated and contain Schwann cells wrapped around the axon that produce the myelin sheath. There are differences in the structure of motor and sensory neurons, though. Let me just summarize that one more time, though. Sensory neuron is any neuron that fires signals inwards towards the central nervous system from your senses. So you sense something, a nerve fires, sends a signal in to the central nervous system. Motor neurons send signals outwards from the central nervous system to the body to actually cause some sort of effect. Now, motor neurons, the cell body is in the central nervous system with the axon leaving it. Sensory neurons, the cell body is not in the central nervous system, it's in the peripheral nervous system. Motor neurons, cell body is at the end of the neuron. Sensory neuron, the cell body is in the middle of the neuron. Motor neuron, the dendrites connect directly to the cell body. Sensory neurons, the dendrites do not connect directly to the cell body. Motor neuron, there is a long axon. Sensory neuron, a short axon. Motor neurons, the dendron is absent. Sensory neuron, it's present. Motor neuron ends at a motor end plate that is just in front of the muscle or the gland. The sensory neuron starts at a sensory receptor, such as the heat receptor. Now, some of our responses, our nervous responses, are automatic. There's no conscious thought involved in them. They're just instant. We need this because, well, sometimes you just have to react instantly. Otherwise, there will be damage to your body. You'd be eaten by a predator. You would hurt yourself. So we have reflex arcs. Reflex arcs are neural pathways which can act on an impulse before that impulse reaches the brain. So, for example, you touch something hot. This stimulates sensory receptors in your skin, thermoreceptors, heat receptors. They will then stimulate the firing of the sensory neuron, which will send a signal up towards your central nervous system. By the way, this means this pathway is known as the afferent pathway. So any signal that goes inwards towards the central nervous system is the afferent pathway. The signal will then connect with some interneurons, possibly in part of your brainstem, possibly a little higher up, and a decision will be made instantly to remove your hand from the sensory stimuli. So a signal will then be sent down a motor neuron. This is the afferent pathway. Efferent pathway means exit. Down to the muscle in your arm, which will be stimulated, and you'll pull your hand away from the heat. So, information from the receptor is sent via the sensory neuron as an electrical impulse. In the spinal cord, the impulse is transferred to the relay neuron. This impulse is transferred then to a motor neuron, either in the spine or the brain. This occurs. Then the motor neuron will stimulate some sort of action. Now, we have many neurons in our central and peripheral nervous system, but we also have a special type of cell known as neuroglia or glial cells. These have a similar origin to neurons, but they're not quite neurons. They help the neurons to perform their functions. That's their purpose. So they're smaller than neurons, but greatly outnumber them. For example, the astrocyte is a type of neuron, and your brain is full of astrocytes. Astrocytes, interestingly, are the first cells that become damaged when you develop Alzheimer's disease. And this is actually quite significant because astrocytes help to protect the rest of the neurons in the brain. So if they are damaged, the rest of the neurons are damaged too. So glial cells support the neurons in various ways. They insulate by providing the blood-brain barrier, the barrier between the brain, the spine, and the rest of the body. Very important barrier, protects you from infection, amongst other things. Protects you by phagocytosis, so some sorts of glial cells, they eat debris and infectious agents that make it into the central nervous system. They nourish the brain by producing something called cerebrospinal fluid, a nutritious liquid that bathes the brain and the spinal cord. They support the neurons, provide stem cells. Stem cells are very important. They are needed to make new cells. Stem cells divide to make new cells. So some provide stem cells and some remove debris. Here are different categories of glial cells, both in the peripheral nervous system and in the central nervous system. In the peripheral nervous system, we can see we have satellite cells. These support cell bodies. We have Schwann cells, which produce myelin sheaths and also secrete neurotrophic factors. Those are basically growth factors that help neurons to grow, mature, and even divide in some cases to produce new neurons. In the central nervous system, the brain and the spine, we have oligodendrocytes, which produce myelin, same as the Schwann cells. We have astrocytes, which I mentioned earlier, very important type of cell. They support the central nervous system. They provide the blood-brain barrier. They secrete neurotrophic factors, such as BDNF, brain-derived neurotrophic growth factor, which allows neurons to grow, make new connections, basically very important if you're learning new tasks. Also we're also involved in ion transport, such as potassium transport, which I won't go into right now. Then we have microglia, which are kind of immune cell. They're scavengers. They basically look for anything bad in the brain and destroy it. And we have ependymal cells. They create barriers between different compartments in the central nervous system. So now I'm going to describe and explain how an action potential, a nervous impulse, is generated, and show you a few graphs that I will not go into too much detail with. I don't want to overwhelm you with information, but I will summarize these graphs. They sort of explain what's going on in a neuron when it's resting and when it fires an electrical signal. So action potentials, what we're seeing here are basically some nerves. Now, nerves are electrically charged. The inside of a nerve is negatively charged compared with the outside of the nerve. It's got a resting potential when a nerve is not firing of about minus 70 millivolts internally. So this negative charge inside a neuron, known as a resting potential, is achieved by pumping sodium out of the neuron. So you have sodium-potassium pumps. A sodium-potassium pump grabs sodium from the inside of the neuron, your body's full of sodium, and pumps it out through a kind of rotating door, like a rotating door in a building, which is actually called a sodium-potassium channel. Sodium is pumped out, and potassium then is pumped in. Now, both are positively charged, sodium and potassium, but more sodium is pumped out than potassium is pumped in. It's on a 1-to-1 ratio. So as this pump operates, in effect, more and more positive charge leaves the neuron. So you end up with a resting potential of minus 70, where you have more sodium outside the cell, outside the neuron, and more potassium inside of the cell. This is known as an electrochemical gradient. Now, when a nerve actually fires, what's happening is channels open that let the sodium flood back in. And as the sodium floods back in, the charge inside the neuron changes, becomes less negative. Now, this stimulates other sodium channels to open along the entire length of a neuron. And as they open, sodium floods in all the way along, like a bunch of dominoes being knocked over. And this basically means an electrical charge flows from one end of the neuron to the other. Now, obviously, you are in a position now where sodium is flooding in, but you've already got potassium inside the cell. So you kind of overshoot. You don't become neutral. You become slightly positive because you've got all that positive sodium coming into the cell, but you already have potassium, which is also positive in the cell. So you become sort of hyperpolarized. You become more positively charged than negative, briefly. And at this point, the nerve can't fire. And then everything is reset. Sodium is pumped out again. Well, first of all, I should say, the potassium is allowed back out. Then sodium and potassium pump starts again, and everything is rebalanced to the resting potential of minus 70. So if a neuron is stimulated, so for example, I touch something hot, sodium channels open, and sodium ions enter the cell by diffusion down an electrochemical gradient. So if I've got a sensory neuron at the end of my finger, I touch something hot, first thing that will happen is little simple sodium channels will open that just let a bit of sodium inside the neuron. This is not enough to make the neuron fire because we have heat around us all the time. There has to be a significant stimulus. So if enough simple sodium channels open and enough sodium comes in, you reach a kind of threshold voltage in the cell. And then this stimulates what we call voltage-dependent sodium channels to open all along the neuron. Sodium floods in, and the electrical charge moves from one end of the neuron to the other. So, first of all, you open up a few sodium channels. Sodium comes in. This depolarizes the membrane, so the inside becomes less negative than it was before. Eventually, an action potential is generated of plus 40 millivolts. And you get an electrical stimulus moving along the neuron. So this is illustrated here a little bit more clearly. So there's a graph and a few images. Take a moment to look at this graph and look at these images so you can see what's going on. We start off with a resting potential minus 70 millivolts. Then simple sodium channels open. Sodium comes into the cell. And the charge becomes a little bit more, well, a little bit less negative. If you get to a threshold potential of minus 55 millivolts, then this will stimulate voltage-dependent sodium channels to open all along the nerve. And you'll get a nervous impulse, a firing of the nerve. Now, as I said, you do have a overshoot because you've got potassium already in the cell. Now what happens is you start to reset. Now you end up briefly even lower than -70. At this point, you're in a kind of refractory period where the ion channels are recovering. Sodium ion channels are closed during this repolarization process. Potassium channels are also closed during this hyperpolarization that I mentioned earlier. What we end up with is a brief period where the nerve cannot fire, just cannot fire at all. So stages in an action potential. In a resting neuron, the membrane potential is polarized at, well, 60 or 70, actually, millivolts. 70 is the one to remember rather than 60. Sorry about the 60 there, but it kind of goes from 60 up to 70. When the neuron is stimulated, some gated sodium channels open and allow some sodium to enter the neuron. The membrane depolarizes until it reaches the threshold potential of -55 millivolts. Many voltage-gated sodium channels are opened and sodium floods in. This is an example of positive feedback. The action potential of +40 millivolts is then reached. The voltage-gated sodium channels close and voltage-gated potassium channels open. Potassium diffuses out of the neuron and the membrane repolarizes. The voltage-gated potassium channels are slow to close. So too many potassium channels leave the neuron, too many potassiums leave the neuron and the membrane becomes hyperpolarized. The resting potential is reestablished by the action of the sodium-potassium pump and the greater permeability of the plasma membrane to potassium. So let's talk a little bit about connectivity now. Neurons send electrical signals down an axon at a speed of approximately 100 meters per second, very fast. How is that signal passed to the next cell? Well, it's passed at the synapse. So one neuron ends, here are the axon terminals. This is the next neuron. Here are the dendrites. There is a signal between one and the next. This area is known as the synapse. And what bridges that gap are neurotransmitters. So here's an image showing a pre and post-synaptic neuron. So pre, the first neuron that fires, post, the one that receives its signal. At the synapse, known as the synaptic cleft, you get one axon terminal releasing some neurotransmitter, which goes across the space, the synapse, binds to a receptor on the next neuron, and stimulates it to fire. Actually, some neurotransmitters are inhibitory, but we won't talk about that right now. Okay, so in terms of synapses, one neuron can actually connect to thousands of other neurons, thousands of synapses in the brain, for example. So one neuron can have thousands of connections with many, many other neurons. And this allows an amplification of a signal and communication between nerves. So there are what we call leak channels. They rebalance in the resting potential. We have voltage-gated channels, such as the voltage-gated channels that let sodium rush into the cell along the neuron when it fires. They need energy, ATP, to drive ions through. And then we have chemical-gated channels. They need neurotransmitters to attach before they open. So different types of channels. Here we have two neurons communicating with each other. So I mentioned there's an electrical signal. So you touch something hot, the nerve is stimulated, you get sodium rushing into the nerve all the way along, and you get a nervous impulse firing along the nerve. Now that nerve could go all the way along your arm into your spine. Now at the end of the nerve, this is where neurotransmitter is released. So the next nerve can be stimulated. How is this accomplished? Well, basically, calcium, which is also positively charged, Ca2+, calcium is pumped out of neurons, meaning you have more calcium in the liquid surrounding the neurons than inside the neuron. This produces an electrochemical gradient. Now, when in an action potential, an electrical charge moves from one end of the nerve to the other, it stimulates the opening of calcium channels. This allows calcium to flood back into the neuron. When the calcium does flood back into the neuron, it binds to synaptic vesicles. Think of them as little bags at the end of the neuron. They are full of neurotransmitter, such as acetylcholine or serotonin or dopamine. When calcium floods into the nerve, it binds to these these synaptic vesicles, and causes them to move to the end of the neuron, fuse with the membrane of the cell, and release the neurotransmitter into the synaptic cleft. This neurotransmitter then binds with receptors on the next neuron, and then some action is performed in that neuron. So, for example, a channel might open, allowing various ions, such as sodium, to move into the next neuron, changing its polarity or signaling gene expression, switching genes on, switching genes off, causing something to happen in the next neuron. For sake of argument, this time we'll just say that the neurotransmitter is going to bind to the next neuron, open an ion channel, which then lets sodium into the next neuron, depolarizes it, and causes it to fire. So one neuron is now fired and cause the next neuron to then fire, which might cause another neuron to fire. So synapses, when an action potential reaches the axon terminal, voltage-gated calcium channels open. Calcium ions then enter the cell. Calcium ions then trigger exocytosis. This means the release of chemicals from the cell. Exocytosis specifically are vesicles containing neurotransmitter. The vesicles fuse with the plasma membrane, the membrane of the cell, of the neuron, releasing neurotransmitters into the synaptic cleft. Neurotransmitter molecules diffuse across the synaptic cleft and bind to receptors on the postsynaptic cell membrane. Binding of neurotransmitters to, for example, metotrophic receptors opens ion channels, leading to depolarization of the membrane. Therefore, the next neuron fires. Neurotransmitters are then removed from the cleft and recycled. Obviously, they have to be removed or the channel would stay open. So there are degrading enzymes that will degrade the neurotransmitter, causing the channels that were opened in the next neuron to close again and everything to reset. And the degraded neurotransmitters are usually taken back up by the parent cell, the presynaptic cell, presynaptic neuron, and recycled. So neurotransmitters are chemicals found in the axon terminal. They bind to receptors in the postsynaptic cell. Receptors can be ligand-gated ion channels. A ligand is just, well, a neurotransmitter in this case, but they're just chemicals that bind to receptors. When neurotransmitters bind to receptor, it opens up, allowing ions to pass through. It may not be ion channels themselves, but may indirectly lead to opening of ion channels. So the receptor might not be an ion channel itself. It might just cause an ion channel to open, or it may actually be a channel itself. Neurotransmitters must be removed from the synapse to switch off the signal. This is an example of negative feedback. Now, here's just a couple of examples of when it all goes wrong, which it can do. The black widow spider produces a toxin called lateral toxin, which causes excess exocytosis of neurotransmitters. This acts at nerve endings, preventing relaxation of muscles, causing technique, constant, strong, and painful muscle contractions all through the body, which can result in the person stopping breathing and dying. Sometimes we use this science, however, to our benefit, depending on your definition of benefit. For example, Botox. Botox is botulinum toxin. It's a toxin that even small amounts ingested into the body will kill you. Very small amounts will kill you. But even small amounts, very, very tiny amounts injected into muscles around the face, for example, can result in the softening or removal of wrinkles. Botox. So this is caused by not enough exocytosis. So basically, what happens is the muscles in your face they're contracting all the time, even if just slightly, and especially when you smile or frown. Now, this constant movement is what's causing wrinkles, really, a bending, a folding of the skin. You don't see it so much when you're very young because you have thick collagen above the muscle. But the collagen does thin as you get older, and becomes damaged, and the wrinkles can get deeper and deeper. Now, to cause these muscles to contract in your face, you release acetylcholine from motor neurons. So motor neurons go to the muscles. Actually, motor neurons go to muscles all around your body and release acetylcholine at the motor end plate. The acetylcholine binds to the muscles and stimulates the muscles to contract. Now, what Botox does is it prevents exocytosis. It prevents the vesicles containing the neurotransmitter, in this case acetylcholine, from fusing with the cell membrane and releasing the neurotransmitter. Therefore, the muscles can't contract, so they relax and smooth out. So synapses between motor neurons and muscle cells, the neuromuscular junctions. Neurotransmitters for all neuromuscular junctions is acetylcholine. So acetylcholine is the neurotransmitter that causes muscles to contract throughout your body. When acetylcholine binds to receptors on the postsynaptic cell, the receptor opens to allow sodium ions in, and the membrane depolarizes, changes its charge. Acetylcholine is then broken down by an enzyme known as acetylcholine esterase. The components are then recycled in the presynaptic cell. So you need this enzyme, acetylcholine esterase, to break down the acetylcholine so that basically the muscle can reset and you're able to then relax the muscle and contract it again. By the way, there are certain chemicals which prevent the action of acetylcholine esterase. Sarin gas is one. This is a lethal gas which has been used in various terrorist actions. For example, one in the 90s in the Japanese underground, the gas was released, people breathed it in, many died because they could not break down the acetylcholine in the muscles. So the muscles contracted and they could not be reset. Integration. A given neuron can receive input from many thousands of synapses, 100 to 500 trillion synapses in the brain. So we've got a lot of connections in our brain. We need them, though, to be able to think. Each neuron may have receptors for many different neurotransmitters. Input can either be excitatory or inhibitory. So a neuron can either receive a stimulatory impulse from another neuron or inhibitory impulse. So we've been mainly talking about stimulatory signals so far. So from sensory neurons, for example. But obviously the brain can't just be stimulated. Parts should have to be inhibited as well. So there are neurotransmitters which are released by certain neurons to calm them down. Basically, one simple example of this, we have our sympathetic nervous system and our parasympathetic. Broadly speaking, sympathetic nerves stimulate, cause the heart rate to increase, the breathing to increase, that kind of thing. Parasympathetic nerves, they calm things down. So for example, when a nerve such as the vagus nerve, which is a parasympathetic nerve, is stimulated, everything calms down, breathing slows down, and the heart rate slows down. We need both. So neuronal impulses can be inhibitory or stimulatory. Okay, so excitatory or inhibitory. Neurons integrate information by summing up input and deciding whether to initiate another action potential. Integration occurs in the cell body. The decision is made in the axon hillock, the beginning of the axon, as to whether to fire or not. So basically, the neuron receives 10 signals saying fire, but 20 saying don't fire, it won't fire. If it's the other way around, it will fire. Okay, so that's a brief summary of how neurons fire. I could go into a lot more detail, and I may do in a future episode, but this was just a relatively brief overview of the process. So thank you very much for watching. If you want to find out more about me, just visit Twitter. and go to @MarkOfTheD and you will find my Twitter account. Thank you very much for listening, and I look forward to speaking to you again. Goodbye for now. Bye.